everyone, welcome to this high-yield review. Look, if you're a medical student prepping for your MBBS exams, you already know that microbiology can feel like this vast, overwhelming ocean of information. But don't stress, because today we're laser focusing entirely on the high-yield blueprint of laboratory diagnosis. We're going to lock in those guaranteed marks by breaking down exactly what you need to know using the best mnemonics and memory tricks to get you through test day. All right, let's jump right in. Here's our game plan for today. We'll start with specimen collection basics, followed by transport and storage rules. After that, we'll spend some time mastering the gram stain, tackle acid-fast staining, and wrap it all up with an exam summary designed to cement this into your brain.
All right, part 1, specimen collection basics.
Getting the right sample is literally the most critical step of lab diagnosis. If the sample is bad, your downstream results are useless. So to effortlessly nail the general principles of collection for your short answer questions, just memorize the acronym CABLES. C stands for collect before antibiotics are started. A is for avoid indigenous flora contamination, which is super important for things like urine and blood. B reminds us that body fluids or tissues are always preferable to swabs. Swabs are honestly just inferior. L is for label appropriately. E is to expedite transport. And S means always use sterile, leak-proof containers. Seriously, just write CABLES in the margin of your exam paper, and you will not drop a single point here.
Now, examiners absolutely love testing specific specimen pairings in clinical vignettes. For example, if a patient presents with sepsis, you have to collect paired blood culture specimens, about 8 to 10 ml for adults, using strict two-step skin disinfection. For meningitis, it's cerebrospinal fluid. For lower respiratory infections, you're looking at sputum or a bronchoalveolar lavage. Pay really close attention to these specifics, because they are absolute must-knows for your multiple choice questions.
Moving on to part 2, transport and storage rules. Okay, so test makers love to target transport rules with trick questions. You really need to notice the sharp contrast between urgent and non-urgent transport. First, you have specimens that demand immediate transport, meaning less than 15 minutes. This includes CSF, sterile body fluids, ocular specimens, and bone. You literally do not delay these, ever. But then you have the exceptions. You can delay transport for up to 24 hours, but only if you use highly specific preservatives. Think boric acid for urine or Carey-Blair transport medium for stool. Knowing this exact distinction is going to save you from so many common traps. Once the specimen actually reaches the lab, temperature is everything. If you can't process it immediately, it splits into 2 storage pathways. You incubate at 37 degrees Celsius, which is body temperature, for blood cultures, sterile body fluids, and bone. But you absolutely must refrigerate at 4 degrees Celsius for stool, urine, and lower respiratory specimens. Why? Because refrigerating those specific samples prevents the rapid overgrowth of contaminating normal flora, which would totally ruin your culture results.
All right, part 3, mastering the gram stain. This is the core diagnostic test.
Over 130 years after it was discovered, this 4-step procedure is still the holy grail of diagnostic microbiology. You start with the primary stain, add a mordant, apply a decolorizer, and then finish up with a counterstain. Now, I want you to mentally highlight step 3, decolorization, because it is the single most critical step in this entire process. We're going to get into exactly why that is in a second, but first, let's memorize the chemicals and timings. To easily reproduce this on paper, use the mnemonic PIDC. P stands for primary stain. That's crystal violet for exactly 1 minute. I is for iodine, which acts as a mordant. You also apply that for 1 minute to form a large diiodine complex. D is that critical decolorization step. You use acetone for a super quick 2 to 3 seconds, or alcohol for 20 to 30 seconds, followed by an immediate water rinse. Finally, C is the counterstain. That's safranin or dilute carbyl fuchsin for 30 seconds. So again, PIDC. Lock those specific timings right into your brain. So why does the stain actually work? It all comes down to the cell wall theory. This is a classic long answer exam question, guys. It's all about peptidoglycan thickness and outer membrane permeability. Gram-positive bacteria have this massive, thick peptidoglycan layer, like 50 to 100 layers thick, with super tight cross-linkages. When you add alcohol, it shrinks those pores, trapping the purple dye inside. Gram-negative bacteria, on the other hand, have a really thin peptidoglycan layer and an outer lipopolysaccharide layer. The decolorizer easily disrupts that LPS, creating large pores that let all the purple dye wash right out. This leaves it completely ready to take up the pink counterstain. And when you look under the microscope, it all comes together. You can clearly distinguish the deep violet gram-positive cocci ranged in little clusters and the pink gram-negative bacilli scattered all around them. Seeing this visually just confirms everything we covered with the cell wall theory. You might also notice some long, slender red bacilli and other stains. Well, that leads us perfectly into our next topic.
Part 4. Acid-fast staining, specifically the ZN hot method.
So when do we use this? This specialized tool is vital when we're trying to identify tricky organisms like Mycobacterium tuberculosis, which have these notoriously waxy mycolic acid-packed cell walls. Let's break down the Ziehl-Neelsen hot method. First, you apply the primary stain, strong carbofuscin, for 5 minutes. But here's the absolute key detail. You have to apply intermittent heating. Heating is specifically required to help that primary stain melt into and penetrate the thick, waxy mycolic acid. Next is decolorization, using a really powerful 25% sulfuric acid for 2 to 4 minutes. And finally, you counterstain with 0.1% methylene blue for 30 seconds. The acid-fast bacilli will stubbornly hold on to the red carbofuscin, while everything else in the background turns blue.
Now, listen up, because this right here is absolute gold for your exams. Examiners endlessly test these specific sulfuric acid percentages because the concentration you need directly correlates to the amount of mycolic acid in the organism's wall. For Mycobacterium tuberculosis, you need the full 25%. For Mycobacterium leprae, it drops to 5%. Nocardia requires just 1%, and acid-fast parasites or bacterial spores need only 0.25 to 0.5%. Seriously, memorize these numbers. They are literally free marks just waiting to be picked up.
All right, part 5. Let's do a quick high-yield exam summary.
Let's simulate the exam environment for a second with some active recall. What is the most crucial step in gram staining, and why does overdoing it ruin the result? Think about it for a second. The answer is step 3, decolorization. If you over-decolorize by leaving the acetone or alcohol on for too long, you will completely destroy that permeability barrier. Even your thick-walled gram-positive bacteria will lose their purple dye, meaning they'll appear falsely negative, and you've totally ruined the diagnosis. Keep that front and center in your mind. So you now have the high-yield blueprint to conquer laboratory diagnosis, but getting the right stain is just the beginning. The real question is, how do these bacteria mutate to resist our strongest antibiotics? We are going to tackle antimicrobial resistance in our next deep dive. Until then, stay curious and keep studying smart.
Thank you.